Welcome back to MCB 170, Society and the Brain. This is Lecture 11, which corresponds to Part 1 of Module 6. The title of Module 6 is Social Influence on Sexuality. In Module 6, we'll try to get a better understanding on the neurobiological, psychological, and sociological levels of sex. So, what is the relationship between culture and sexuality? According to thinkers like Michel Foucault, sex influences culture as much as culture influences sex. Sex influences culture just as human experience in general influences culture. Take eating, for example. Eating is an essential human experience, and the experience of eating has been elaborated into the fantastic cuisines of the different cultures. Similarly, sex is a basic human experience, and sex has been elaborated into various different cultural aspects. But why would society, through culture, religion, law, and in other ways, strive to influence the sexual behavior of individuals? Well, Michel Foucault was a famous French philosopher who helped us understand power relationships in society. And Foucault pointed out that many sexual relationships are based on power relationships. And many laws develop to protect weaker people in society from stronger, more powerful people in society. For example, in most countries, it's illegal for adults to have sex with children below a certain age. And that makes sense. The laws were there to protect children who have less power from adults who have more power. But other laws don't seem to protect anybody. Up until a few decades ago, same-sex marriage was illegal in the United States. In 2015, however, the Supreme Court ruled that same-sex marriage should be legal in all 50 states. We'll discuss the sociological and cultural aspects of sex more in Part 2. In Part 1, I'd like to focus more on the neurobiology of sexuality. And that begins with the sex hormones. Here we have the structures of progesterone, testosterone, and estrogen, the three major sex hormones. They are all steroid hormones. That means they are all derived from cholesterol, and they all have this basic cholesterol structure, this four-ring structure. They differ in the nature of the atomic groups that are bound to that four-ring structure. The sex hormones are produced in the gonads, the ovaries and the testes. The gonads, the ovaries and the testes, are part of the endocrine system. The endocrine system is basically the hormonal system. The endocrine organs produce and secrete hormones. This diagram illustrates the endocrine system. It includes the gonads, the ovaries and the testes, also the adrenal glands, which we talked about in previous lectures. The adrenal glands actually sit on top of the kidneys here. Other endocrine organs include the pancreas, thymus, and the thyroid and parathyroid glands. The secretions of these endocrine organs are under the control of the pituitary, and the secretions of the pituitary are under the control of the hypothalamus. We talked about the hypothalamic pituitary adrenal axis in the control of cortisol. The hypothalamic pituitary gonadal axis controls the secretions of the sex hormones from the gonads. Before we discuss that, I want to point out a very interesting fact. You might think that males produce only testosterone and females produce only estrogen and progesterone, but that isn't true. Males also produce estrogen and progesterone, and females also produce testosterone. So both sexes have all three sex hormones. They have all three gonadal hormones. Interestingly, the gonadotropic hormones from the hypothalamus and the pituitary that control the secretions of the gonads are also the same in males and females. Let's take a look at that. Here is the hypothalamic pituitary gonadal axis in diagrammatic form. Males and females have testosterone, estrogen, and progesterone. In males and females, neurons in the hypothalamus secrete gonadotropic releasing hormone, GnRH, into the anterior pituitary. GnRH stimulates the anterior pituitary to secrete luteinizing hormone and follicle stimulating hormone into the general circulation, LH and FSH. LH and FSH then stimulate the gonads to secrete the sex hormones testosterone, estrogen, and progesterone. The testes produces mostly testosterone, but it also produces some estrogen and progesterone. The ovaries mainly produce progesterone and estrogen, but they also produce some testosterone. Interestingly, the adrenal glands also produce a small amount of the 3 sex steroids. And the sex steroids, just like the stress steroid cortisol, exert negative feedback on their own production. The sex steroids are best known for the development of secondary sexual characteristics during sexual development. As you all know, there's a surge of the sex hormones around puberty. That surge of sex hormones enables the development of our secondary sexual characteristics, and you all know what they are. They account for the sexual dimorphism between male and female human beings. Sexual dimorphism is the difference in size and shape between males and females of the same species. So we know that the bodies of men and women are different. What about the brains of men and women? Are they structurally different? In humans, it turns out that there are 3 peak periods of sex hormone levels. During puberty, which we just mentioned, also the perinatal period, that's the period just before to just after birth. Just before birth and just after birth is the perinatal period. And during the first half of gestation. Gestation is the time between conception and birth. It's the whole period during which a baby is developing in the womb of its mother. The gonads are formed during gestation, during the first half of gestation. So the gonads appear very early in the development of a fetus and start to produce the sex hormones. So the sex hormones are around pretty much since the beginning. And structural differences between male and female fetuses in their brains are also around since very early on in development. They're apparent between 4 and 7 months of pregnancy. So there are differences in the size and shape in certain brain regions in male and female human beings early on. And those regions are primarily the supraoptic, ventral medial, and paraventricular nuclei of the hypothalamus. Okay, this is interesting. The sexually dimorphic brain structures, the brain structures that are different in male and female human beings, are in the hypothalamus. They're hypothalamic nuclei. These are little subnuclei of the hypothalamus. Neurons in the hypothalamus, subnuclei in the hypothalamus control basic functions like drinking, eating, aggression, and sexuality. And these 3 hypothalamic nuclei that are involved in sexual behavior are different in size and shape between male and female human beings. We know a lot about the neurobiology of human sexuality, but we know a lot more in animals because we can do experiments on animals. In rats, there's a critical period in development from a few days before birth to a week after birth. This is the perinatal period, during which sex hormones can influence the formation of sexually dimorphic structures. That happens in humans too. It's a little bit different between humans and rodents. Hormones have lesser effects at other times. That's true for humans too. The sex hormones have huge effects during their surges, during gestation, the perinatal surge, and the pubertal surge. They still have important effects in the adult, but not as great. Once developed, sexually dimorphic regions of the brain can be activated by sex hormones to produce sexual behavior. This is very interesting. It's observed in the rat, it's observed in the human. During development, the sex hormones can influence the development of organs, including the brain, including parts of the brain. And it can ready those organs and those parts of the brain for later activation by the sex hormones. So the sex hormones essentially ready certain brain structures for later activation by the sex hormones. Here are three important sexually dimorphic regions in the rat brain. Let's do the last one first. The spinal nucleus of the bulbar cavernosus is larger in males because it contains motor neurons of penile muscles. So the SNB is controlling the muscles of the penis, and it's kind of obvious why that would be bigger in males than in females. The anteroperiventricular nucleus is larger in females than in males. APN neurons project to PVN neurons, paraventricular nucleus neurons, and promote the secretion from PVN of oxytocin. We talked about oxytocin a lot in the last module. It's a hormone important for maternal behavior and also important for pair bonding in adults. The APN is larger in females than in males. The sexually dimorphic nucleus of the preoptic area, SDNPOA, is at least 2 times larger in males than in females. And it's necessary for the expression of mounting behavior. We'll talk about mounting behavior in a minute. But mounting behavior is only one aspect of sexuality. The sexually dimorphic regions are necessary for sexual behavior. In adult males, destruction of the medial preoptic area, otherwise known as the SDNPOA, we just talked about it, eliminates copulation. The males will still approach receptive females, but they just won't mount and copulate. Similarly, destruction of specific areas of the brain eliminates specific aspects of sexual behavior in adult females. So in both males and females, sexual activity is not controlled just by one part of the brain. It's controlled by many parts of the brain. There are several parts of the hypothalamus that are involved in different aspects of sexual behavior. And other parts of the brain from the hypothalamus and the spinal cord and the brainstem all the way up to the prefrontal cortex are involved in sexual behavior in human beings and probably in other animals too. So let's look at the basic sexual response. Sexual activity in most animals and in almost all mammals involves mounting and lordosis, back to front copulation, in which the male's back is concave, the female's back is convex, the male mounts, and the female produces lordosis. It turns out that we know a lot about lordosis, and the study of lordosis is fascinating, and we're going to go into it in some detail because it's going to show you how different parts of the brain interacting with the hormone system all contribute to the control of sexual behavior. It's not just happening at a low level. It's initiated and ultimately consummated at a low level, but many other parts of the brain are involved in its control. But essentially, the lordosis response is a reflex. This is the basic structure of a reflex in the context of the flexor withdrawal reflex, which is an example of a reflex arc. What is the flexor withdrawal reflex? It's a very, very familiar reflex. It produces withdrawal of a limb when the extremity encounters a painful stimulus, maybe sharp or hot, crushing, some other painful stimulus. What happens? Well, if you poke me in the hand with a pin, I will flex my limb. I will withdraw my limb by contracting my flexor muscle. How does that work? It originates with receptors, sensory receptors in the skin. These can be touch receptors, pain receptors, heat receptors, pressure receptors, receptors for kind of intense, noxious stimuli. We'll activate certain sensory neurons. And here you see these little action potentials, these little nerve impulses, waves of ionic current travel down the sensory neuron and the sensory neuron will then activate interneurons. And the interneurons will activate motor neurons. The motor neurons will activate the muscles of the flexor muscle, and the limb will be withdrawn. The presence of the interneurons is very interesting. You might wonder, why don't the sensory neurons contact the motor neurons directly? Well, whenever there's an extra step in a biological process, it's there for regulatory purposes. It turns out that neurons coming down from the brain can influence the interneurons and either enhance or suppress reflexes. Reflexes can be kind of complicated. They occur at the level of the brainstem. So here's a cross section of the brainstem. Sorry, not the brainstem, the spinal cord. Reflexes are found in the spinal cord, which is the lowest level of the nervous system of the brain. It's considered separate from the brain. And you see here the withdrawal reflex for the right foot. Here you see the right foot stepping on a tack. The pain receptors signal through sensory neurons, which contact interneurons, which contact motor neurons, which activate the flexor muscle of the right leg and pull the right foot away. But that's not all that's happening. You see this rather complicated set of interactions occurring within the spinal cord. The cell bodies of neurons live in this darker region of the spinal cord. All these cell bodies are interacting together through dendrites and synapses. Not only does this reflex activate the flexor of the right foot to pull the right foot away, it also is going to relax the extensor muscle so the extensor won't fight the flexor and allow the leg to retract. But on the other side, it will relax the flexor and excite the extensor so that the left leg extends and supports the weight of the individual while the right leg is withdrawn. So it can be kind of complicated circuitry, neural circuitry here in the brainstem, in the spinal cord, excuse me, in the spinal cord. We'll get to the brainstem in a minute. The lighter part of the spinal cord is white matter. It's axons, fiber tracts that go up and down the spinal cord to and from the brain. The pain experienced by the foot, sensed by the pain receptors in the foot and conducted by the sensory neurons into the spinal cord, will actually make contact with other neurons that will send that signal up to the brain, so the brain knows that there's been a pain in the foot. But conversely, axons can come down from the brain to influence this reflex, mostly at the level of the interneurons. And it can suppress a reflex, for example, when you really want to withdraw your foot or your hand, but there's some reason why you shouldn't. For example, if the object that you're touching, which is causing you pain, is very valuable and you don't want to just drop it. The lordosis reflex is also a basic reflex. It's initiated by stimulation of Ruffini touch receptors near the hindquarters. So a scientist named Ruffini discovered these touch receptors, which aren't necessarily pain receptors. They respond to lighter touch, and they can activate the lordosis response under certain circumstances. The sensory afferents sensory neurons relay the signal from the touch receptors to interneurons in the spinal cord, which in turn activate the motor neurons that elevate the hindquarters and produce lordosis. However, the interneurons are modulated by descending input from the brainstem, which determines whether or not the lordosis response will occur. So the lordosis response is a reflex on the local level in the spinal cord, but it's influenced a lot by descending axons from the brain. So here's a diagram. Here again is a cross-section of the spinal cord. Here's the dark part, the gray matter in the spinal cord, which is all the cell bodies and dendrites and synapses. The white matter is all the axon tracts, myelinated axon tracts coursing up and down to and from the brain. Here's a picture of the sensory neuron coming in from the mechanoreceptors. What's a mechanoreceptor? Mechanical receptors, receptors that respond to mechanical stimuli, pressure, heat, not heat, that'd be temperature, pressure, pain, touch. Sensory fibers come into the spinal cord, activate interneurons. The interneurons might be arranged in a complicated way to coordinate activity between the two sides of the body. Then the motor neurons project out to the back and leg muscles that are going to actually produce the lordosis response. Notice that sensory signals can go up to the brain to inform the brain that the Ruffini endings have been stimulated, but influences can also come down from the brain to modulate this reflex. And here we see the spinal cord coming down from the brain. This is the rat brain, front of the brain, back of the brain. Notice that neurons in these regions are projecting down the spinal cord to the hindquarters, to the region near the hindquarters that's going to mediate the lordosis response. Where do these influences come from? They come from the medullary and midbrain reticular formation and the central gray matter. What's the reticular formation? Reticulum means net in Latin. So the reticular formation seemed to early neuroanatomists like a net-like, web-like agglomeration of neurons. We already talked about nuclei in the brainstem. Nuclei are compact bunches of neurons. We talked about the raphe nuclei, for example, that synthesize and secrete serotonin. We also talked about the locus ceruleus in the ventral tegmental area that synthesize and secrete noradrenaline or dopamine. They were distinct clusters of neurons. The reticular formation are the neurons kind of between those distinct clusters of neurons that mediate very primitive functions. They've been around a long time in evolution. What is the midbrain and medulla? Well, the spinal cord comes up into the brainstem. The brainstem is the lowest part of the brain in your head. The brainstem has important nuclei in it, as we just mentioned, like the raphe nuclei. And in between those nuclei is the reticular formation. And the medulla, pons, and midbrain are just different sections of the brainstem. The medulla, pons, and midbrain are just different sections of the brainstem. You don't have to remember that in detail. You just have to remember that neurons in the medulla and in the midbrain project down the spinal cord to modulate and control the lordosis response. Neurons in the central gray do that too. What's the central gray? It's kind of related to the reticular formation. It's gray matter, cell bodies, dendrites, synapses, near the brainstem, above the brainstem, in a kind of a diffuse region that's going to mediate very basic functions like sexuality. So the central gray, the midbrain reticular formation are going to project to the medullary reticular formation. The medullary reticular formation then projects down the spinal cord to modulate the lordosis response in the spinal cord. Now, these three regions, the midbrain and medullary reticular formation and the central gray, are in turn under the control of the ventral medial hypothalamic region, the ventral medial nucleus of the hypothalamus. So this whole response is being coordinated by the hypothalamus. The hypothalamus in this case is not only controlling the secretion of hormones, it's also controlling neural activity in the central gray and the reticular formation, which in turn is going to influence activity occurring down in the spinal cord. Interestingly, the ventral medial hypothalamic region is under the control of the sex hormones. This is a diagram showing the results of experiments where experimenters made estrogen radioactive, and they injected that into a rat, and they could later detect the radioactivity and see where the estrogen bound in the brain. And they found that estrogen activates neurons, can influence neurons in many different parts of the rat brain. Here's the front of the brain, back of the brain, especially down here in the hypothalamus. The preoptic area is part of the hypothalamus, and here's the ventromedial hypothalamus. This is the part that we're talking about. This is the part of the hypothalamus that controls the lordotic response in females. How does estrogen do that? The lordosis reflex can only be initiated in the presence of the female sex hormones, estrogen and progesterone. The sex hormones exert their effects by acting on neurons in the ventromedial hypothalamus, as we just said. This action occurs on long and short time frames. This is a specific example of how sex hormones can prepare neural structures for later responses to the sex hormones. Remember that hormones, their basic function is to tell cells to make more of some proteins and less of certain other proteins. Their basic function is to modulate gene expression and protein expression. On the long time frame, on the developmental time frame, estrogen enters the neurons in the VMH and activates intracellular steroid receptors. We talked about that before in the context of cortisol. Steroid receptors like cortisol, like the sex hormones, are essentially fats. They pass right through cell membranes and they can activate receptors inside the cell. So on the long term, estrogen is acting in that classic manner by activating intracellular receptors. The activated intracellular receptors go into the nucleus and modulate gene expression and protein expression. The activated estrogen receptors cause the neurons in the VMH to synthesize various proteins, including intracellular and membrane-bound progesterone receptors. So estrogen, during development, goes to neurons in the VMH, and it tells them to make certain proteins, including progesterone receptors. And these progesterone receptors are not only intracellular, classic receptors, but also membrane-bound receptors, receptors that sit on the cell membrane and can respond to progesterone very quickly in the circulation. Then on the short time frame, like just before sexual activity, progesterone can activate VMH neurons by activating those progesterone receptors, okay? So this shows us how estrogen primes the VMH for later stimulation by progesterone. So let's put this all together, starting with the hormones. Steroids, and in the adult mainly progesterone, activate neurons in the ventromedial hypothalamus, which activate neurons in the central gray matter and midbrain reticular formation, which activate neurons in the medullary reticular formation, which project to interneurons and motor neurons that control lordosis. That's starting from hormones to thalamus to this gray matter in the brainstem, down to the spinal cord reflex that controls lordosis. The modulatory pathway, this pathway coming down from the brain, acts synergistically with sensory input from Ruffini endings to produce lordosis only when touch stimulation and sex hormones are present together. So you see that on the one hand, lordosis is a reflex, but it's a reflex that's under control of the nervous system and the endocrine system. Why is that? That's because reproduction is a critical function and it wants to be regulated carefully by the brain and the endocrine system. Okay, let's switch to males. We don't understand mounting as well as we understand lordosis, but it turns out that we do understand erections pretty well. Why? Because medicine for erectile dysfunction has become a big business. What causes an erection? Erection is due to increased blood flow into the corpora cavernosa. What's that? It's a network of blood vessels in the penis. So here's a diagram of the penis, and here's all these blood vessels. The penis is highly, highly, highly vascularized. There's lots and lots of blood vessels in it, and some of the blood vessels are elaborated into this kind of spongy-like structure that absorbs blood and causes erection. Blood flow into the corpora cavernosa is due to relaxation of the smooth muscles that usually restrict penile blood flow. Okay, what are smooth muscles? Well, there are three types of muscles: striated, cardiac, and smooth. Striated muscles or skeletal muscles are the muscles of your skeleton. They're the ones that help you lift weights. Cardiac muscles are the muscles in the heart. The contractions of the heart are due to contractions of the cardiac muscles. Smooth muscles are found in other organs, like in your gut, in your stomach and intestine. They're responsible for the squishing around that helps you digest your food and makes funny stomach noises. Smooth muscles are also found in your blood vessels. Smooth muscles control blood flow in most of your arteries. Normally, smooth muscle cells in the penis are contracted. Normally, smooth muscles in the penis are contracted, and they prevent blood flow into the penis. So they prevent an erection. Erection occurs when those smooth muscles relax. What causes the penis smooth muscles to relax? Certain neural projections secreting transmitter onto those smooth muscles. They work through metabotropic receptors, which activate G proteins, which lead to the production of cyclic adenosine and guanosine monophosphate. These second messengers activate other proteins that eventually cause smooth muscle relaxation, which leads to blood flow into the corpora cavernosa, which leads to an erection. That's shown in this diagram here. Certain transmitters, neurotransmitters or neurohormones, activate receptors. Those receptors are metabotropic. They're G protein coupled. If you activate this G protein here, they're calling it G sub S, you increase the production of cyclic GMP and cyclic AMP, those two second messengers. High concentrations of cyclic GMP and cyclic AMP result in relaxation of smooth muscles. You do not have to remember the details of this diagram, but you do want to remember about cyclic AMP and cyclic GMP, these important second messengers. Conversely, other neurotransmitters or neurohormones can act through metabotropic receptors to activate different G proteins, which increase concentrations of different second messengers, which lead to contraction of smooth muscles and prevent an erection. Remember cyclic AMP and cyclic GMP, because these are going to be important when we try to understand the mechanism of action of drugs that produce erections. But what are these transmitters that occur naturally? What are the transmitters that can facilitate or suppress an erection? Well, here's a list right here. This list of transmitters, hormones, and other factors contracts the smooth muscles and eliminates or suppresses an erection. And this set of transmitters and hormones and other factors relaxes the smooth muscles and causes an erection. We haven't talked about these too much, except for these three: noradrenaline, serotonin, and dopamine. The big three monoaminergic neurotransmitters. They can get into your body too. They get into your blood also, and they can affect erections. It's interesting that noradrenaline and serotonin both prevent erections, while dopamine enhances erections. Okay, what about Viagra? What about drugs that produce erection? How do they work? Sildenafil, sold as Viagra in the US by Pfizer, works by inhibiting phosphodiesterase 5. Phosphodiesterase 5 is an enzyme that breaks down cyclic GMP. So here's our second messenger, cyclic GMP. When the concentration of cyclic GMP is high, it leads to relaxation of smooth muscle cells, increased blood flow into the penis, and erection. Viagra and other medicine for erectile dysfunction essentially blocks phosphodiesterase 5. This leaves cyclic GMP intact, unaffected, and that cyclic GMP will relax the smooth muscle cells. Sildenafil and other phosphodiesterase 5 inhibitors are used to treat clinically significant erectile dysfunctions of any origin. Well, what causes erectile dysfunction? Decreased blood flow, decreased nerve supply to the penis can prevent or impair erection. So phosphodiesterase 5 inhibitors were developed to treat that condition. But the non-prescription use of Viagra and other PDE5 inhibitors is increasing rapidly, especially among older men. Well, in older men, there can be disruption of blood flow, disruption of nerve supply, and leading to erectile dysfunction. So lots of older men are using these drugs to treat that condition. Sometimes, however, younger men who have normal blood flow and nerve supply to the penis also experience erectile dysfunction. This is called psychogenic erectile dysfunction. What causes that? Why would a guy who has normal, healthy blood flow and nerve supply to his penis fail to get an erection when he wants one? Well, a group of researchers actually investigated this. They showed erotic video clips to men with and without psychogenic erectile dysfunction. Young men who normally get erections, and young men who have impairment of their erections. They found that men with psychogenic erectile dysfunction have greater activation, red, in the ventromedial prefrontal cortex, caudate nucleus, superior parietal lobule, and posterior cingulate cortex. Men without psychogenic erectile dysfunction, or any kind of erectile dysfunction, have greater activation, blue, in the insula, hippocampus, and anterior cingulate cortex. Now, these results are complex. They're a little bit difficult to understand. We've talked about many of these regions before. We've talked about the insula being involved in disgust, hippocampus involved in stress and memory, anterior cingulate cortex involved in conflict detection. Why would these be active in men who don't have psychogenic ED? It's not clear. We also talked about the caudate nucleus, it's part of the pleasure circuit, and the prefrontal cortex. The prefrontal cortex is the seat of reason. It's also the main structure in our controlled processes. Why would the prefrontal cortex be involved in psychogenic erectile dysfunction? It's almost as though the prefrontal cortex is controlling the erection away, probably in some psychologically maladaptive way. And one of the treatments, it turns out, for psychogenic erectile dysfunction is cognitive behavioral therapy, which we talked about in the context of depression. Through cognitive behavioral therapy, a man can better understand his and guide his controlled processes, hopefully to prevent and treat this condition. I'll end there.